So I have to declare that my talk has nothing to do with dengue, so apologize because I could not make it yesterday and this afternoon, so Edmund says, But it's still aligned with immunity. The 4 keywords are immunity, lessons, bat, and inspiration. Okay, so that's kind of what my talk is going to be. Okay, so very briefly, it's a short talk, so I have to go fast, so I will present some of the bad facts. For those of you in the audience that have heard my talk, I apologize. And then I think really my career started with bat-borne viruses, okay? But today's talk is really to demonstrate that, your career can go, beyond what you thought you were going to finish up. So now we're really getting into this bat-inspired, new approaches to medicine and health in general. Okay, so again, most of you have seen these slides. Bats are very unique as a flying mammal, occupies around 20% of the living mammal species, but really focus on the last 3, right? So, I learned after I studied bat-borne viruses, bats actually, relatively speaking, has a very long lifespan, a health span, and then low rate of tumorigenesis. Last but not least, this is really I start my career, is identify bats as a major reservoir for all the emerging viruses, and many of them are very deadly. Okay, so really relevant to the later talk is about stress and the stress sort of resilience. So when you think of stress, some are like disease infections that you don't get normally. But for bats, they get this stress every day. Bats are nocturnal, so they're flying out nighttime. So their metabolic rate goes up to 30-fold of the base rate, and their heartbeats can go all the way to 1,000 beats per minute, and body temperature goes to up to 42. It's not every bat species. There's over 1,000 bat species, so the different one has different temperature. But I always say, imagine, you close your eyes, tomorrow you can fly. It's an advantage that nobody can compete. But you will not survive for a few hours because with this kind of, a biology change, you will be dead within an hour, So bats obviously is a mammal, right? So that they have, acquired this over at least 60 million years of evolution. So we don't know the ancient bats 65 million years ago when they just acquired fly, how much survived, but they evolved to really resist this kind of stress. Okay, so my career started accidentally, went to Australia, and in the lab we discovered that the first bat-borne lethal virus, the Hendra virus that I named, and then I have been associated with the major bat-borne viruses, and all the way to SARS-CoV-2. So bats as a very unique reservoir for emerging zoonotic virus, is not being debated anymore. 20 years ago, we still debate whether bats are unique in its ability to harbor viruses, but that now, I think it's beyond the debate. Really, the debate is what makes bats special, Is that ecology, is that genetics, is that, the evolution, adaptation to fly, or unique immunity? I think all of these contributed. But today we'll focus on, the immunity. So, as I said, 65 million years ago, I'm not evolution biologist. I think evolution is always a random event followed by selection, right? So why this class of mammal acquired flight capability, we cannot explain. But clearly, fly offers a lot of advantage, but also a lot of stress, as I already illustrated. So there's a huge evolution pressure, to adapt to this. Then when you can fly, you will be exposed to different environment and different, pathogens, just like in this audience. So you have extra sort of stress from the exogenous. So internally, you flight-induced stress, but also exogenously. So there's a lot of adaptation, and I can spend hours to give you examples of what bats have we don't, as a flying mammal. But the key thing is these 3 unique features, right? They are the viral reservoir, so I basically contribute to, pioneer this field. But this is a new area, I did not know when I went to bat research, and, but now we become sort of very familiar with that. So on the right is the review paper basically summarize all the mammals. You have an index, right? So the body size versus, lifespan. If you normalize as a 1, only 19 mammals really go above that, and 18 of them is a bat. So clearly there's something special. And last but not least, anecdotally and now with experimental data suggests that bats are just resistant to all sorts of disease, from cancer to diabetes to heart disease. Okay, so I don't have the time to go through it, and for those of you interested, we do have a review a few years ago, summarize this balance of what we call defense versus tolerance. So if we illustrate that, but that's like 3 years old now. So in those days, we have illustrated that some of the innate immune genes, like the interferons, ISGs, H proteins, are constantly expressed at a high basal level than human and mouse. Okay, they are battle ready, but if you have a virus or you have other stress, they don't go as high. Certain of the genes, immune response or inflammation don't go as high. So they're a bit more balanced. That's kind of our main conclusion. So I use inflammation as an example for to illustrate the difference between bats and humans, basically. So I think Scott Neer is sort of the founding pioneer of this field. I remember I was at one of the only inflammation meetings. I'm not an inflammation sort of biologist, but I attended a meeting in Sydney. And he basically declared to say, inflammation is the roots of all human disease, especially when you're 50 years and above, okay? So what that says is that, so I just artificially put a list there, aging, inflammation plays a very important role in the aging process. COVID-19, we know why certain people have severe, not others just asymptomatic or very mild. Inflammation play a very important role. And Zika virus is hot right now, so inflammation also play in pathogenesis. There's a strong evolution sort of pressure, for bats to dampen the inflammation response. But that, for most people says, there's a danger because inflammation is protective if it's the right level of inflammation. What Scott Neal said basically is inflammation is highly conserved from low sort of a level class mammal to all the way to humans because it's part of the protection mechanism, the innate immunity basically. But when you have over-inflamed, that's the roots of all disease or pathogenic, right? So the balance is the key. So for bats, for somehow, maybe because the flight, induced the stress, these are the triggers for inflammation, but they fly every night. So if every night you have a strong inflammation, that's kind of disastrous for a species. So this, I think, is the main driver. And that inflammation is regulated by many, many different mechanisms. And in the last maybe 2 decades, inflammasome, become a hot area. So inflammasome is really, in health and disease, has been documented in many different areas. And then, really, I always said that our lab is only one lab, right, in only the last 8 years. So we have found many different mechanisms that bats have adopted, and we don't, and to really try to keep the inflammation in check, prevent over-inflammation. So many different mechanisms, but I will demonstrate, I think, the better in the next slides, sort of diagrammatically that. So this is the inflammasome that highly conserved across mammals. We have it, a mouse have it, human have it, right? So you have a different pathways, to go in, and you have a sensor. Okay, you have an adapter, and the downstream, then you go all the way to R1 beta and the caspase. So we have identified at least 6 different mechanisms from priming to really the activation, inflammasome activation, that bats have a dampened mechanism. All right, so at the sensor level, we identified 2. One extreme is the double-strand DNA sensor, which is the M2 molecule, is completely deleted. Bats are the only mammal does not have M2. Others, like a mouse, have multiple, so there's a gene family. And this is not in one bat, this is in 1,400 species of bats. None of them have the M2. So it's a very strong nectar selection during evolution. And then the adapter molecule, the ASK1, I will give you an example. And the downstream, we have the caspase and R1 beta. So I will give you 3 examples at 3 different levels, at the sensor level, at the adapter level, and at the downstream sort of. So at the sensor level, M2 is deleted, so it's easy to understand, But for NLP3, which is a very important sensor, and there's lots of anti-inflammatory drugs use this as a target, and at least 4 or 5 still in clinical trial, but without much success. So what we discovered is that NLRP3 in bats are there, highly conserved. The sequence is relatively similar to human mouse, but it's much less active. It's there. It can still play a function, but it's not as active. So, these are published data. I don't have the time to go through it. Just to illustrate the implication, what that in the context of infection immunity. So if you have the human PBMC and the bat PBMC, you infect it with most coronavirus. That data was published before SARS-CoV-2, but SARS-CoV-2 did the exact same. So if you infect this, you stand by the viral load, the similar, so they all get infected, virus replicate. But then if you check the activation of the ASK1 complex and downstream inflammation, that in human, unfortunately the lighting, but you have to believe me that, okay, how to go back? Red arrow? Okay. All right. So, the activation of the ASK1 or the inflammasome activation is 10 times higher than bats. So bats get infected, but does not go as high of the inflammasome activation. The next is the adapter in the middle, right? So all of these sensors have to go through this adapter and then activate downstream caspase and IL-1 beta. So that's the ASK molecule. And there's a decoy. So ASK has 2 domains. One is a binding, one is a catalytic. So ASK2 basically is a decoy. You only have the binding domain, does not have the catalytic domain. And only human has it before we discovered that, or human and primates have it. Mouse, pigs, cow, name it. No other mammal have it. And we found that bats have it. Not only that, human is like it's left over. It's like our appendix, right? It's really there, but not functional. So ASK2 is really never turned on unless some organ is really damaged by inflammatory activation, and then they activate. But still, if you use the same protein quantity to assay the activity, the bat ASK2 is much more active than the human ASK2. Not only that, the bat ASK2 is constantly expressed. So that's a very, really, so we learned the lesson from this very sort of interesting study, is that first, evolutionarily, that this the molecule human primates and bats have it, but bats the only one constantly express that. And then it's really play a role in attenuate, all the inflammasome sort of activation. And we have now a patent on a molecule that batize a human ASK2, use 4 residues from bats, now become active, can be a drug, So just to illustrate, sometimes if you had the right discovery and the right thinking, so it's really lucky because the mouse does not have ASK2. So to make a batized mouse, transgenic mouse with a bat ASK2, you don't have to knock out the background ASK2, you just put a mouse ASK2. So that one gene from bats, and you make a transgenic. And then you try to, again, we always use the infection in my lab as the first kind of demonstration. So if you have a lethal model of influenza infection, then all dead by day 10. And this bat ASK2 is 50% protective. I have to say my lab is very, very bad in making bat molecules and into mouse. So the expression level is not good. So as you can see, the infection level is similar, that it's really the inflammation has been dampened. And we extend that to 2 other RNA virus, the bat virus and also the double-strand RNA virus and also polystrand RNA virus. It's the same results. So basically that, that mechanism assure that bats, explains why bats such a good reservoir is they can allow the virus to replicate but does not go over immune response. And then the downstream now is caspase-1 and also the IL-1 beta. Again, I don't have time to go through it. It's a beautiful story. We have 3 different bats, basically, and then one basically lost the caspase-1 activity, and the other one basically, lost the IL-1 beta activity. And 3 bats have 3 different mechanisms, but the end results are the same. If you get an infection, for example, human get really high, inflammation, where the other 3 are dampened. So it's a huge. So the lessons, I focus on the lessons, right? So I only explain, the dampened inflammation, in the context of infection. But my lab now has to go way beyond that infection. Actually, 80% of my research is infection, not infection-related anymore. So metabolism, we feed the sugars to bats. They don't never develop diabetes. And then, cancer, senescence, aging, and skin and neurologic disease that to do with it. And we just graduated world first bat cardiologist, PhD, So that's kind of interesting. And I don't have time even to go to heat shock, because that's another whole area. So basically, it's a bat-inspired medicine and health. That's what we are proposing. And in Singapore, we are sort of leading the world because we have every component. So we have a breeding colony, we have the omics, we have the cell line, organoid. So we can go all the way to really human application. People always challenge me to say, are you in dreams to learn from bats and actually benefit human health? 20 years ago, or 15 years ago when I was defending the grant, I said, yes, I can do it. But you need to give me 10 to 15 years, right, to achieve that. So now, sort of, we achieved that. So this is a telegraph, we have a commentary, Bats, unfortunately, people always thinking that as, bad. But I never said it's bats' fault to give the virus. Bats and the virus has been coexist for millions of years. It's the human activity. So we always ask, is bats good or bad for human, as a society? And then finally, Nature Biotech, 2 years ago, have this, says bats biotech takes flight, basically to say it's been humans' dream for 100 years, try to utilize bats to benefit human health. Myself and Emma Tilly, Emma Tilly from UCD in Ireland. So we really started, and then now there's a company, $100 million company, Paratus, using our S2 as the first sort of target to go to a human anti-inflammatory, new broad anti-inflammatory. And the board of the company just approved a human clinical trial next year, 2026. So if everything goes well, we will have a first bat-inspired broad anti-inflammatory drug for human use. Okay, so with that, I'd like to thank my Betty MD PhD student. Another way for me to build a confidence that we're along the right track is these doctors, right? Future doctors come to do a PhD using bats. That's, the first one was very brave, Mate. Now he has become a tenure track professor himself. So that gave me extra confidence. So I thank my students, and I thank all the, everything looks like a bat now, right? So when we have a parties, we will have bat. And then this is the photo sent from my US friends, So it's a bat cave. It's not bat, it's a cave. But from inside, if you take a photo, it looks like bats. So everything looks like a bat. Thank you. I don't know if you have any question. As I said, my one is very high level. Oh, Antonia has a question.
I have a question. No, very nice presentation.
Thank you.
So, while you were talking, I was thinking. So, innate immunity is necessary also to have a very good, let's say, adaptive immunity.
Yeah.
Have you ever tested vaccine in bats and see what's happening?
Yeah, so that's a good question. I know. I was thinking I was going to sort of from this company, the ProImmune. So there's two arms, right? The way Yang has gone great detail about, the antibody versus T cells. So we can only do, unfortunately, it's not, we don't want to do T cells. We don't have the tools. So I challenge the ProImmune, to give us the tools to do bat T cell. So nobody has developed tools to do T cell response in bats. Everybody sort of go for grant and don't get money, so do not. But in terms of the antibodies, we have done lots of this. The last 25 years, we have been doing that. The conclusion is clear that bats' adaptive immunity, the antibody arm, is very weak. Okay, so
it makes sense.
One example is the Hendra virus has been surveyed bats, horses, and humans for the last 25 years. In bats, the neutralizing antibody. So neutralizing is a much more accurate assay because it's a secondary antibody independent. If you do ELISA, people always argue you don't have a good anti-bat antibody, right? You use a protein AG. So we use neutralizing, the same assay, use human, horse, and bat. Bats, you'll be lucky to get a 1 in 120 titer. Horses, we get 1 million. Humans, 20,000. But to answer your question directly, we also did a vaccine challenge studies in bats. And within 6 months, antibodies all gone, and the peak titer never as good as in mouse or ferrets.
Yeah, so
but T cell is really the key thing. I think that's why I'm excited. We already work with ProImmune to see if we can finally get a T cell to work for bats.
Thank you.
All right, thank you.